Well, Dr. Guy Setton, thank you so much for joining us. Everyone, this is the CEO of GemmaCert. Let me just get the right screen here. Now I'm looking at you. Great. Guy, thank you so much for giving us your time today. I really, really appreciate you being here. I am a huge proponent, just so you know, Guy, I'm a big proponent of the future of this technology in the market. And I don't think that there is anyone better that I could talk to really get a good feel of where the technology is at and what the future potential is for the cannabis space. So just thank you so much for taking the time to come today, Guy. Really appreciate it.
Thank you very much for this opportunity, and I appreciate it very much.
Well, Guy, you know, I've got some questions that I want to ask you. First of all, I want to acknowledge we are broadcasting from unceded native territory here in Vancouver. Guy is joining us from Israel, and it is quite late for him. So again, Guy, you've done some sacrifice to be here. I very much appreciate it. Okay. First question that I wanted to ask you, Guy, is I want to talk about the historical background behind spectrometry, okay? I want to talk about not only the era of Newton to start with, but some people before Newton as well, right? 1600s is when this practice begins. Am I correct, Guy, or am I totally out to lunch here? Somebody earlier?
I mean, you know, people have been studying the impact of, or trying to figure out what's inside of herbal medicines for hundreds, if not thousands of years. Because if you go all the way back to traditional Chinese medicine, then you have various, hundreds, if not thousands of different flowers, varieties, which are used for their therapeutic benefits. And there's been a constant or continuous effort by scientists, professionals, amateurs alike throughout history to try and decipher what makes those plants work. Spectroscopy, which we are using in GemmaCert specifically, has been widely used, I would say, pretty much since the 70s and 80s of the previous century. And that has been used really in food and beverage, pharmaceuticals, dietary supplements, agriculture. I mean, there's not an industry today which doesn't use spectroscopy somewhere along the supply chain, primarily for ensuring quality, knowing what's coming into your facility, knowing what's going out of the facility. Because at the end of the day, you are selling a product, doesn't matter what that product is, you're selling a product to the consumer. And if you want that consumer to come back and buy the product again, they need to be assured that they get the value for their money, that they trust you for what you're selling them, they trust the label, they trust the brand. If it's on the recreational side, if it's something that is emotional, then they want to make sure that every time they buy that product, that they pay money for that brand, that they're getting the same experience. If it's someone suffering from a particular ailment, they want to know that every time they buy the product, they get the same efficacy. And that's where quality obviously comes into play. And spectroscopy is an effective means for ensuring quality.
So why don't we do a little bit of a rewind? How does this technology work? What is spectroscopy? How does light tell us about whether or not cannabis or any product is at a certain standard of quality?
Exactly. So basically every active compound out there is, at the end of the day, it's a molecule. And a molecule will react when it's hit with light of a particular wavelength. And so when you are lighting up a sample, or let's say you shoot light on a cannabis flower, then THC molecules will react in a particular, you can notice them on a particular wavelength. And CBD molecules are on a different wavelength. And then by the reaction itself, the intensity of the reaction, you're also able to use sophisticated algorithms, basically mathematical and statistical tools, to estimate, how much of those, how many molecules or how potent that is, it's in the flower. So it's a combination of spectroscopy enables you to identify and to quantify the molecules. If there's nothing there, then you won't have any impact on the wavelengths.
So I'm very interested by this concept that, because you said something that really triggered a lot of thought in my head right now. There's very few industries related to pharma and food that do not have spectroscopy currently at some point of the supply chain.
Correct.
So really, you're talking about grain, you're talking about any pharmaceutical product, fruits, vegetables. Is it in the meat industry, Guy? Even like, are they going to utilize it? Yes, even in meat for
it's used pretty much. There's not a single industry out there that doesn't have it somewhere, because the beauty of spectroscopy is that it enables you to get real-time results without affecting the product itself. You don't have to. You're basically not even touching the product. It can. In some industries, the spectrometer is on a mass production, like on a conveyor belt, and product is passing at very high speeds. I can tell you, I was recently in a date processing facility where they had high-end spectrometers there. I think they were analyzing 9-10 dates per second. And being able to quantify the moisture level and other parameters. So it's everywhere. Because
it's starting to dawn upon me, because I mean, right now, this technology has only really started to scrape the surface when it comes to the cannabis industry, right, Guy?
Correct.
I mean, it's what would you say right now that is the number of LPs, any producers worldwide, what would you say is the number of them that are currently using spectroscopy in their production SOPs?
I would say today, you're probably talking less than 10%, 5% of the market. And one of the reasons or challenges that we face in the market, and I'm sure you've heard it as well, is that I'll tell it to you this way. When I came into the market the first time, which was back in 2014, 9 out of 10 people, professionals, okay, I spoke to, said, I don't need to do any testing. I know exactly what I'm growing. I've been growing this for 5, 10, 15, 20 years. It's stable, and therefore I don't need to test anything. But when the business became more and more regulated, and expectations rose and oversight increased, they realized that you can't just say, I know what I'm growing. You need to actually provide evidence. You need to give confidence to your customers, to the regulator, to your partners. And so slowly but surely, there's more awareness for testing. But even today, in 2021, you still have this misconception of many licensed producers that they say, well, if let's say I grow in this perfectly stable, controlled environment, I control the lighting, I control the temperature, the nutrition, everything's controlled in my facility, therefore everything it comes out the same. And that's wrong. That's a wrong assumption, because what interests us is not the fact that the parameters in the greenhouse are stable. We want to know that the chemical composition the final product is exactly the same every time. And there's so many things that can go wrong in the process that you cannot simply rely on the fact that you had a stable temperature or you knew exactly the nutrition or lighting that you were controlling throughout the grow process. And so today there are more players out there who are understanding the importance of testing all throughout the process and not simply depending on good luck or, you know, on chance that it will come out the same. Yeah.
It is surprising to me. I will say this. Look, I'm a grower from before the days when we had testing available to us at all. 2001, when I moved here, you couldn't really get your plant tested at all. And I was a patient and I really wanted to know what was in there. Back then, we didn't know if we had 1-to-1 or, in fact, there was very little old school CBD dominant plants we found out later through testing. And it's very difficult for me to believe. See, this is my opinion. If you started growing in recent years, like 10 years recent, where testing has been seen as a pain in the butt to all of these, oh, it cost me so much and I don't always get the result that I want, and oh, I freaking hate testing. But for those of us that come from before the days when we were blind, we wanted to know so bad how to select my phenotype. For me as the breeder and as a grower, that was my number one thing, is I want to know which one of all of these versions from my seed run biochemically, not just in terms of me liking it more. So it's always a combination of those two factors. In fact, for the breeder, it's how it feels olfactory and in terms of consumption, combined with the biochemical analyses that guarantees that what I'm experiencing is actually reflective of a reality. And it's very difficult for me to believe that growers would be like, you know what, this thing, see, this is the thing, a lot of people already know that it works, because there's a lot of folks out there right now that don't believe spectroscopy works on cannabis for some weird reason. Like, it works on everything else. This technology function, this is the thing, because it's not the same as an HPLC, a lot of people believe that it doesn't work. But that's, I think, part of the misconception that I want to throw out today, that technologies can have different methods of obtaining data, and that data, based on the fact that you're obtaining it from that piece of technology, can be used in specific ways. Low cost, repeatability, the fact that you don't destroy the sample. It has different applications than HPLCs. HPLCs are going to destroy the sample. It's not that easy and cheap to replicate the test over and over again. So it's very difficult for me to believe that folks are like, no, I don't want the capacity to test at an exuberantly low cost in-house before I go to the HPLC. And again, I feel strongly that that resistance has a lot to do with either being old school and having worked closely with HPLCs and just not wanting to believe that you can replicate that with a spectrometer, or having come in early into the industry and not having had the experience of being blind and not actually seeing the value and the strength that it gives you. Because, I mean, frankly, Guy, you have to be somebody that writes SOPs and maybe more than once to really get it. I mean,
yeah, it's totally, I mean, it's such a big misconception to think that that you cannot have an industry like all the other industries where spectroscopy lives side by side with the HPLCs and the GCs perfectly well, complementing each other. The HPLC, as you mentioned, it has its place in the lab. And it has its functions for example, for mandatory regulatory testing. But when you're in the field, and field, I mean, it could be in the greenhouse, it could be in the warehouse, it could be at the retail point in the back office, it could be in a clinic or a pharmacy. You're not going to wait, hours, not to mention if you have to send it out, wait 6-10 days or even more to get a result back. And then you get a result for a product which no longer exists, and you're talking about a natural product where there's natural variants. So that's why they don't. It's two different things. And if you even when you speak to also specialists in other industries and also regulators, they will tell you spectroscopy is a reliable and credible means for quality assurance for testing. I mean, for, you know, the FDA permits, you know, the biggest pharma companies out there to use spectroscopy on their production lines before they release medication into the market. You know, I'll give you this small anecdote just to you know, explain the best way. One of the drivers for bringing spectroscopy into the pharmaceutical industry in mass production was Pfizer. Pfizer came out with Viagra in the 90s. Demand was shooting out of the, you know, out of the roof, and they came to the FDA and they said, "Listen, we cannot supply demand if we need to wait days for some laboratory to send us results. We need to have something in real time give us assurances that we can release the product into market to meet demand." And the FDA said, "Fine, use spectroscopy. If the results come in fine in real time, and that's enough of a credible risk mitigation system to let the product go to market." And the rest is history. Today, every pharmaceutical plant in the world uses spectroscopy, and HPLCs are still used. It doesn't contradict.
And I think, you know, Guy, we're going to get more into the specifics of how your technology works in a moment, because the intersection between AI and these types of measurements is highly interesting. But I think that, like, that's part of what I think leads people astray in all this, that they believe that there's an inherent competition between laboratories and this type of technology. And I like, as a person that's used the technology and continues to use laboratories, I have to say that, like, as well as, I'll be frank, Guy, I've just gotten a lot into how the actual technology works and the data points that it takes and how it combines them. And it's obvious that without laboratories, technology like this simply cannot work. It cannot. And the thing is, plants continue to, we breed new varieties, we create new chemotypes of old flavors. Like, you know, nowadays there's CBD dominant death bubbas out there. There's all this stuff. We're going to continue to have to go back to the labs over and over. You can't just stop going to the labs, because these are images that are being compared by algorithms that require input. They require data input and verification to ensure that the images are calibrated to the right type of result ranges. So without people diligently testing at labs, without labs being there, none of this could have happened, and it cannot continue to happen in the future. I really feel that, look, spectroscopy is here to stay. We all need to come to terms with that. And because, you know, anybody who's still on the bandwagon that says that none of these devices work, you just haven't used the right one. I'm sorry, I have to say that to any of you out there. They do work to a certain extent for specific applications. They can't give you the readings for all your terpenes and for every range of cannabinoid that's out there. You can't do that yet, but it can give you reliable readings as to the ratio of THC and CBD. I've tried to trick your machine, guys, so many times. Give it the mix one, give it the mix ratio, and then give it the THC right after, and then switch it to CBD dominant. And every time, it might not be exactly the same as the HPLC results, but it's following it closely, and it's providing me with enough understanding about my ratios that when I'm out in the field, as somebody who's a breeder, and I'm trying to get rid of anything that has an excess of 3% CBD, trust me, this is just because, look, if my cultivar has 1 to 2%, I'm happy. Rounds off the high, really works well for my consumer. The moment that I get CBD in excess of 3 to 4%, it starts nullifying my THC dominant effect. And then even if I have 28%, it doesn't feel like a 28% anymore. Especially, of course, the right terpenes need to be there, right? Point being that I think, you know, Guy, I want to talk more about specifically how GemmaCert works, because it's a bit of a unique device that doesn't exactly do what everyone else out there is doing. So talk to me about intersection between images, AI. How does this baby work?
Good question. Well, when we embarked on solving the challenge of testing a single flower bud without having to touch it, without having to grind it. But we basically said we want to take a single flower bud, and we want to be able to use spectroscopy to measure the amounts of THC and CBD in there. And all the spectroscopy solutions that exist in the market take a single point analysis of a given area on the sample. And therefore, when you're dealing with, let's say, a homogeneous active pharmaceutical ingredient, white powder, then anywhere you take a reading, you'll get the same result because it's homogeneous. It's usually a synthetic active pharmaceutical ingredient, no challenge. But when you're dealing with a natural product, specifically cannabis, where the most simple image analysis of a flower, a heat map, will show you that the trichomes are not spread evenly across the flower. In fact, there are areas in the flower bud which have no trichomes whatsoever. And so if you're using single point, it's like Russian roulette. You may hit a point where you got a trichome, you may hit somewhere there's no trichome. So until we showed up, people said, you know, there were other technologies and still out there that say grind the flower. And even grinding is not necessarily a recipe for a homogeneous sample. So what did we do? We said we need to be able to take multiple scans of the flower. And in order to take multiple scans of the flower, you need to know where to navigate. And again, a flower bud is not a perfect shape with straight lines. And for the spectrometer to always be at its peak performance, it needs to have an optimal distance from the flower. So you got these cameras, which are in a sense, they're the eyes of the spectrometer to make sure that every time it is in a different location at the optimal distance before it scans that particular point. And in our devices, for example, in the optimal setting, we're taking 24 different scans. Obviously, you can tweak that and go down and shorten the analysis time, but at 24 different scans, you're taking 24 different readings in the flower, always at the optimal distance, and all that data is going up into the cloud, and that's where it's being compared to the reference database. And that brings me back to what you said just a couple of minutes ago. Our reference database, as you correctly pointed out, is based on thousands and thousands of samples that we have tested on our devices and then on our HPLCs. We have HPLCs in our lab which are running, and we cross-reference to the HPLC. And only data which has been cross-referenced with the HPLC is the data which actually goes into the cloud and is the basis for the algorithms which are doing the comparisons. Now, and just one more note, there's also a misconception there in the market. An HPLC is also comparing. Unlike, it's not using a reference database, it's using a standard chemical. You are comparing the extraction that you made of the cannabis flowers to the reference standard that you purchased for THC, which could be 99% purity, whatever, but it's also an act of comparison. And let's not forget, the HPLC, as you pointed out, you need to have a skilled technician operating it. You need to prepare the sample. You need to interpret the results, and you destroy the sample. And you got also hazardous solvents that you need to dispose of afterward. So it's not the most practical solution.
So you test in-house, but you also work with third-party laboratories that will provide you with verification of samples that are inputted into the mainframe guy? Or is it all?
No, because we want to make sure that all the data that we use is uniform and meets our standard. Only data which we have tested ourselves through our protocols on the HPLC and on the GemmaCert devices, that's the data that goes into the reference database.
Understood. So it's in-house verified. Protocol is always the same.
Always the same.
Protocol is always the same. Testing protocol is always the same. Okay, that's an important point. And then AI algorithms basically compares those images to everything that's in your data bank. You say that there's well over how many data points would you assume are in your data bank right now? Over thousands, you would say, right?
Yeah, I mean, we're talking, I mean, if you think of, we've tested, I think well over by now, you know, in the 20-30,000 flowers at least, and each flower you're testing it 24 times before you put it into the HPLC. So it's big numbers.
Okay. I think that like that's something that I'll just say this much. I've bought other devices that I'm not going to say which ones that use this technology, and I was just not impressed with the accuracy at all. The why it wasn't accurate, like trying to figure out why it wasn't providing me with repeatable analyses. I always figured, well, it has a lot to do with the way that I'm holding my sample, which field I'm in. Am I indoors? Am I outdoors? Did I hold it too tight to the device? Did I poke it too deep? What human error was there? And what logically made sense? Not only did I get better result, like closer results on my GemmaCert right away, but I also thought about it like this device has to be put on a completely solid surface that is completely flat and in a room that's not too hot. All of these factors are actually stipulated for you, because I couldn't figure out how the hell they could give me, how they would think that this handheld device could give me accurate samples when I'm literally taking it to Mexico with me and like stabbing something at like 100% humidity at like 35 degrees centigrade outside. It's not the same. It's not like, no. Because I haven't worked in the technical department of a laboratory ever, but I've worked for laboratories and I understand how to market them and how to talk about what they do and explain that to a client in a way they can understand. It doesn't make sense to me that a handheld device, unless there was some other additional, maybe again, some AI-based component, but even then, if you're not collecting the data in a standardized manner, in a basically
closed,
in an enclosed, exactly, it has to be an enclosed area, because it doesn't, otherwise how could a spectrometer really do its job effectively? That doesn't mean that, by the way, anybody out there that's bought a different unit, that doesn't mean your unit is garbage. I'm just saying to you that it doesn't really make sense to me. And I've personally, as well as many of my colleagues, have experienced repeatable results, meaning that I'll take the same cultivar that I tested 3 days ago, and I kept it enclosed in a container that does not have a lot of dead space, and I take it back, and my AW measurements, as in my water activity measurements, which I want to talk about in a second, Guy, are pretty much where they were, and my THC content and CBD content are reflective of what I tested before, within usually a percentage point or two. Really, it's very, very close if I'm testing the same sample. I could not get that same result with any other device that I've owned in the past. And I have owned some that I know for a fact were just random number generators, because I like put oregano in there and it was still giving me like 20% THC. So if you put oregano in the GemmaCert, it'll be like it's 0%. You know, it's not going to give you any readings on there. So right off the bat, that'll be inspiring. But Guy, talk to us about water activity, because look, CBD and THC, I could already get that from most of my labs. I wasn't getting water activity readings. And you know what, Guy? I found out that my curing methodology is giving me AW readings that are very sensible, that will be within the range of what you want. I usually end up at a 0.56, 0.57, right above the 0.55, which is the way I like it. I like it not like too gummy, not too crispy, you know, right where it needs to be, in my opinion. But Guy, talk to me about this water activity. How important is water activity to the cannabis industry? And most of these people checking MC, is that enough? Moisture content, is that good? you think?
Moisture content and water activity are in a way, they're similar, but they're different. And what I mean is that water activity is an indication of how many water molecules are basically roaming around freely in the flower, ready to hook onto something. And when you have too many water molecules that are free and looking for something to hook on, that's a ripe field for mold or other mycotoxins, and you want to avoid that, obviously. So your objective is, as you point out, is really to have a AW score, or water activity score, of, let's say, between 0.55 to 0.65. And if you fall in that range, then you know that your product is safe for transport, safe for storage. You don't have to worry about mold becoming an issue there. Obviously, you need to make sure you're putting it in good storage and check it frequently, but you got a good product on your hand. If it's below 0.55, you probably have a very dry product. It'll probably also obviously have a low moisture level. It could be already, it's usually a sign that the trichomes are degrading. So there should be doubts about the potency as well. And if it's too high, if it's above 65, so that's usually a very moist flower. And you have a very, very high risk of that flower being infested or infected with something. So that's and again, this is ABC. This is basic practice in the food and beverage industry. When you want to move product from one place to another, you need to make sure the water activity is safe.
And look, I know that some of you that are joining us work for LPs currently, and you're scratching your head right now thinking, why aren't my SOPs reflective of any of the stuff that they're talking about? First point, they actually have paid close attention to the type of information that Guy has said, but they've oftentimes chosen to take a safe and easy road. And I've mentioned this in previous episodes, where a lot of companies, they do the standard MC content for grain in Canada, and they utilize that as their storage protocol for cannabis. What is that? Guy, you might know this. It's 10% MC. So 10% MC translates in terms of AW. Let's talk about this a little bit, because again, for me, taking your stuff past the 0.57, 0.58 starts to get a little crazy. People that are like, oh, I'm going to pack it right at 0.64 because I want my stuff to be super goopy. I'm like, you know what? They're like, oh, but I did a microbial test and it came out at 0. I'm like, yeah, for the sample you submitted to the laboratory. Okay. That's one of the things that this type of technology one day is going to help us fix, because currently, correct me if I'm wrong, Guy, you cannot test for aflatoxins. That's, remember, those of you that are watching us, that's a fancy word for mold residuals. Okay. So we can't currently test with aflatoxins in the device, but Guy, do you think that that's going to happen soon?
We are looking at various ways, also using image analysis to try to come up with solutions for that. The problem here, or the challenge here, is that a lot of these issues or events, if you want to call them, in the flower, you need to detect them when it's at the level of accuracy levels of parts per billion. And when you're trying to bring a product which is affordable, which is fast, and can be used by anyone outside the laboratory, you don't have that capability today at those price points. So there are some features that which are not within our reach, like pesticides and various contaminants, where again, all you need is a trace element at parts per billion to rule out the entire batch or whatever. Yeah. So we're not there, no.
We're not there, but I definitely think that by having the AW reading, you can have way better protocols and SOPs, because look, not every site is going to have somebody that's like me that does everything by tact. this is what I found out with your device guy, because I wasn't checking for water activity, is that my method that I was implementing naturally was already lining up with where I was supposed to be in terms of the measurement of my water activity. But oftentimes people are not going to have somebody that's been growing the same plant for 10 years and knows exactly how, because depending on what time of year is it, some people don't have curing facilities that are completely, perfectly, hermetically sealed. And you're going to have fluctuations in your SOPs depending on what type of year, time of year it is. Namely, for example, in Kelowna, up in the mountain in the summer, it's going to be dry as hell a lot of the time. And then during rainy season, that moisture is going to change depending on whether or not it's raining outside during the days that you are curing. So it will extend your curing protocol depending on how hermetically sealed your space is. That all of these variables are diminished when you have a standardized protocol that says that you're just going to continue to test this bud until you get to the 0.56 that Adolfo likes, and then you're going to stop. That way you don't have to have somebody that knows really well, based on tact, how it needs to finish, based on how all of the variables happened in that batch. You know, was it grown exactly the same as the previous time, et cetera, et cetera, et cetera. So yeah, for me, water activity, look, because people aren't checking right now, this 10% thing, this 10% MC, they're like, look, the SOP, we're not going to check water activity because we don't have a machine that does that because they cost too much. Okay, that's what people think. Not so. It's worth it because MC is not good enough. And it ends up leading people towards these lazy SOPs, standard operating procedures. They start becoming, oh, you know what, if I set it at 10, then the chance, because that's so dry, that's basically typically going to be below the 0.55 for most cultivars. Remember that AW, as in water activity, varies depending on the morphology of your bud. Okay, namely, a very tight bud, you want to crisp that up a little bit more than a very elongated wispy bud. Elongated wispy hazes, you want to maybe not take them to the 0.56, you want to leave that one at a 0.58, 0.59, because you know that the nature of that morphology will create more airspace inside the packaging. And remember that dead airspace inside of packaging creates terpene dissipation, as well as the redistribution of all of that MC, of all that moisture content, not only amongst the fibrous portion of the bud, but also amongst all of the extra air that's in your packaging, right?
That's exactly, I mean, that's exactly the point, is that when you test for only moisture content, so you get a result of how much water there is in this product. But if you want to know what is the risk of that water reacting, you need the water activity.
And guys, I'm going to tell you something that you may be aware of it because you've got the right tools to know, but very few people have a lot of experience with true hazes. Okay, I'm from Mexico, so I have experience with true hazes, and I spent time in Jamaica. I went looking for hazes, and I found them, and I grow them on the regular. And I'm going to tell you something right now, that if you have a standardized SOP for your MC or your AW on a haze as compared to a Kush, again, Kushes, you do not want to leave them at a 6.0. You don't want to leave them at 6.1. You don't want to leave them at 6.2, because in the context of a Kush, that is above the water activity that actually favors the product in terms of its preservation capacity, but also how it plays on the palate and how it feels to the touch. When a Kush is too wet, it like doesn't come out of your grinder. It's so hard to roll. It doesn't burn well, might burn a bit darker, right? So having a Kush that's anything that's very dense with a water activity that's even slightly too high, you've ruined your product, in my opinion. And you know what's very likely to happen? Because you think you're within the bounds of regular water activity, but you're not thinking about morphology. And what happened is you put it into the packaging, and then it starts to develop that, like, especially Kush does this, that like dank basement smell that's also like kind of like, it's like peppery and like almost like fermentation together, which is not the desirable aroma profile that you want to bring out of your Kush dominant lineages, right? So haze on the other side AW needs to be closer to the 6.0. You do not want to be close to the 0.55, as in the case of Kush, where you want to be 0.56. Haze, what you want with haze, what's the most desirable part of haze? Is it the fact that it has a crazy amount of THC? No, it's all about the monoterpene preservation. So knowing about water activity and preserving a high content of water activity when you package allows you to deliver that elongated cultivar the way that the customer wants to consume it, still with the citrus, still with the fresh cut grass aroma combined with the citrus, sweet to the nose. That's what they want. Remember that sweetness, whether it's limonene or terpenoline based, it's going to dissipate so quickly if you've got not enough moisture in there. So water activity, I'll just pass it back to you, Guy, because I've got more questions, but you can see my passion for moisture, Guy, because I've really realized how much moisture is the make it or break it aspect and factor of whether or not the nose is there on my cultivar when my consumer receives it, so it's really important to me, man. Pretty passionate about it.
No, I can certainly feel that and see that, and that's, by the way, that's one of the reasons why we are, since all our devices out there, and today we have devices operating in 50 countries, so we're really experiencing a whole variety of different cultivars and experiences. But that, since all our devices are connected to the cloud, every time we have a new feature, an improvement, an optimization, model update, we immediately upload it to everyone's devices like this. Right, actually today we released CBG.
I was hearing about that, man. Amelia was telling me about that. Guy, that's big, man. That's big. As of today, I can use my device to check for CBG today.
Exactly. Yeah.
Whoa, man, that's awesome. I didn't even know about that, man. That's so cool.
We so if you open your device today and you'll see the app updated, you will see there's a result now for CBG. So we're constantly working on things.
So, Guy, I always wanted to know. So you know what I'm going to be doing now that you told me? I always wanted to know at what rate the CBG in the early stages of growth converts to THC, and the rate of conversion and how that varies depending on cultivar and genetic. Oh, man. Tell me you're going to do THCV soon. Tell me it's coming, please.
Trust me, we're working on a lot of things.
Okay, that's exciting. That's really exciting, Guy. That's super exciting. I feel like I did the reveal show today. I didn't know it was a reveal show. This is surprising, man. That's awesome.
You see, you got it live. The world knows about it.
So for those of you that are not that familiar with CBG, or maybe you don't understand why Adolfo is so freaking excited right now, CBG is the mother molecule. It's the molecule that the plant produces initially, from which THC, CBD, CBN, everything else is derived. So having a good handle, this is the other thing, right? CBG is a desirable particle. So maybe it starts to create a whole nother wave of production where maybe I start breeding a plant, for example, that is no longer desirable to harvest at the peak of maturity. Maybe Adolfo now breeds a plant that's specifically dedicated to CBG production. And I happen to know that CBG sometimes peaks right before you enter the peak stages of flower. So CBG actually becomes quite concentrated in some cultivars and expresses itself in some cultivars better at earlier stages of life. What does that mean? I could harvest this plant quicker, flip quicker, get more rounds out, sending those unmatured flowers over for extraction with the current amount of CBG that was in them. Of course, depending on whether or not I'm able to determine which one of my phenotypes is capable of doing this at a rate that is actually feasible financially. So now I've got the ability to do a whole nother aspect, a whole nother program of R&D, Guy. Today can start in my site thanks to this, man. This is big. This is big. Yeah, this is cool. Also, if you're scratching your head, what is CBG used for? CBG is a lot less psychoactive than THC. It's kind of CBD-ish. And there's been a lot of research that's looked into PTSD, different types of psychiatric conditions. And they found some positive results there. There's anti-inflammatory properties, antifungal properties, antibacterial properties. Like most cannabinoids, it has a bunch of different functions to it. And we've only begun to scrape the top of what this compound can do. There's like no CBG product in the market right now, but it's coming. So
it's coming. I mean, we are. I mean, from talks that we're having with customers all over the world, there's certainly an effort on the way to get those high or CBG dominant strains into double figure CBG potency levels. So I'm sure there's going to be something coming soon in terms of dominant CBG varieties on the market.
yeah, the highest I'd ever seen was a 12.
Yeah.
And I know that I've heard whispers of a 16er in, you know who's doing it. It's the Spaniards, of course. That whole crew of people that's around the CBD crew, they've got the heaviest, stable, rare cannabinoid genetics, man. That whole crew of people is like hats off. Doctor, I don't remember what the PhD is that's associated with them, but the publications that they put out and the work that they're doing is just exceptional. Guy, you have a cool job. You get to work with all these cool companies all over the world, eh?
It's never a dull moment, I have to say, in this industry.
How well adopted has this technology been more on your side of the world, Israel and Europe? How is it going down there?
Well, I have to say that you know, as I said, we sell the device now in 50 countries, and no specific market you can say is really dominant or overshadowing all the others. I mean, even though the US market is the biggest in terms of numbers, it's obviously a very important market for us, and so is Canada. But we have devices from you know, Tahiti to, we have in Bariloche and in almost every corner you can think of, Hawaii. So basically it's all over the place. I mean, Israel is a hub for cannabis research and development, both on the breeding side as well as delivery systems, the precision agriculture. So for us, being a company in Israel, it was a very, very comfortable platform to develop the technology, because we had access to licensed producers providing us with material to build a reference database. Academia in Israel, Israeli universities, we specifically came out of the Hebrew University of Jerusalem, which, as you may well know, is the home of Professor Meshulam. So, you know, we're walking in the corridors of faculties, which cannabis is not some weird thing, but has been common discourse for the past since the 1960s. So it's a very, very fertile ground upon which to bring innovation to the cannabis market, and that's been the issue here, or the advantage that we've had being an Israeli company. And today, yeah, I mean, it's also, we have devices and I would say around 35 licensed producers in Israel using our technology.
I think that is this going to lead into our next question? It's so shocking to me how hard it has been for the industry to adopt this, because I mean, I'm really into the stars and the planets, and knowing what this technology has done. I mean, not just for, like you say, the food industries that are quite relatable, but I mean, this technology is used by NASA, and it's used very effectively to do amazing things with it, and it's essentially the same components, the same type of concept. I really am interested to hear your answer to this. Where do you think that the most disruptiveness, the most change will happen based on the adoption of this technology in the cannabis industry? Where are we going to use it? How is it going to change things?
Well, I'll tell you where I think the ultimate game can be, or where the holy grail of this technology. When we are using spectroscopy to test cannabis, we are obtaining the raw data is what we refer to as the unique spectral fingerprint. of that particular flower specimen. And it's very similar to your fingerprint. It's unique. And our vision is that we will be able to, together with licensed producers, to basically bring products to market where we have in our database the specific spectral fingerprints which are reaching the customers, so that when the customer consumes the product, they can afterward, by simply scanning a QR code, answer a few questions. And based on the feedback, you will be collecting real world evidence on what someone experienced, and you will be able to correlate it to the fingerprint of the exact product which they consumed. And here's where machine learning, artificial intelligence comes into play, because when you're talking big numbers of real world evidence, you'll be able to go back to the breeders and say, listen, if you can formulate or breed a genetic makeup which brings on a consistent basis, regular basis, a fingerprint similar enough to this fingerprint which we are now handing over to you, you can be guaranteed that your consumers will have that same experience over and over again.
Yes.
And that's, I think, the ultimate game.
So you're talking not only about changes in the way cannabis industry works, but how it relates also to the way we're using the data and the way that it relates to consumer and patient output, and how they basically report that they've reacted. Has it helped? Has it not helped? Right. So Guy, you're taking in some of this data as well, and you're starting to mix it into the data pool. You're starting to do that, eh? So basically, reports, first-hand reports as to how a particular fingerprint made you feel, and then starting to mix that into the data pool a bit, and maybe providing. So it looks like you might have several services to provide in the future with that data, eh, Guy?
Yeah. I mean, it's a longer term project, and it's to some it may seem science fiction, but all the tools are available today to make it happen. And I'm sure that and then I mean I do see a scenario where you could actually walk in one day into a dispensary with your smartphone, scan a particular product barcode or QR code, and your profile is on your smartphone, and you would get, let's say, a green light which would say this bud will be great for you, yellow light, try it, no harm, red, stay away from that bud. And that's something that's possible.
Yeah, if you've identified a clear aversion, for example, a lot of people do not like terpenoline. No terpenoline dominance for a lot of people exacerbates sometimes anxiety conditions or different type of, some people just don't like it, I definitely think that there's a future there. I also think that, it's not just that there's a future there, this is the type of work that needs to be done, regardless as to whether or not we end up at this ultimate place where we can always differentiate for sure. And I do have to say that there's a lot of companies out there that try to pose as if they can do it. For example, Leafly is the top of my list, that they try to act as if they're a data crunching company based on data points that are able to tell us potential effect, but they have not collected any data in regards to the actual biochemical constituency of the specific
product. Exactly. There's a there's a big black hole there.
It's obvious.
And it could be, it could be data worth for entertainment, but it's not something that you can really reach a scientific conclusion.
No, because they've collected basically input from multiple people that have tried Blue Dream, and there's multiple different phenols of Blue Dream. Even if you have the actual genetic line, single genetic line, multiple phenols, which one did you get? Well, there's one that's mango dominant on the aroma, another one that's blueberry and fresh cut grass on the aroma, and another one that's really quite dull, that's to be discarded, that should not be grown at all, in my opinion. So which aroma profile did you get from the actual genetic line? Do you have the actual genetic line? Matters less than if you have the right biochemical profile in terms of preference. Some people don't get that part. They think, oh, if it's closely related to the Kush line, it's always going to be the best choice. No, Kush is one factor that can help the consumer connect with their product, but combining that type of knowledge with biochemical contents and never forgetting that sometimes cultivars from different ancestries can have similar biochemical content, you know? So especially when you're talking about lots of wild type plants that will very commonly recede to a myrcene / beta-caryophyllene dominant aroma profile, very dirty, very earth. And that tends to happen a lot with genetic drift on most wild cultivars. So a lot of them end up smelling very similar, even though one of them comes from India, the other one comes from Mexico, this one comes from the other part of the world. So,
Yep.
so ancestry only takes us so far. This type of information and having it, imagine, this is the other thing, Guy, this is the thing that drives me crazy, that batch numbers are sometimes relevant to batches of varying sizes. So batch number is sometimes relevant to 2 kilos and sometimes is relevant to 100 kilo, okay? So what a device like this could do for us consumers is that you no longer have to do the single batch testing for the full 200 kilo. Now your jar can tell you quite specifically what was in that specific jar. Because remember that not all growers grow like me and Mike, my partner that grows with me. We growers that are well experienced, you produce your plants so that it has mostly only top cola. They all grow very similar to each other at similar height, and we do not use the smaller cola, the smaller buds as included into the jar. So if you buy something that I grew, you're never gonna get a bunch of small bud and then one big bud, because like Guy was saying, those small bud, they don't contain the same amount as that big bud, okay? That's not the same quality of product. Sometimes it is if the genetic is leaning towards producing small bud, and then there's one large bud anomaly, that's a little bit different, but that's not typically the case. Typically the case, if you're seeing a bunch of different sizes buds, that's different quality of product, especially considering how much green matter there is in the smaller bud. If it's a small bud, it already had less THC, but now it has more green matter, which is going to dilute the amount of trichome expression that occurs on that fibrous piece of bud. And now your numbers are way off. Now you're consuming something that's 12%, whereas your container said 22%. So I think this is the thing that for me, as a cannabis sommelier, somebody who's very obsessed with knowing exactly what I have in front of me and being able to train my senses to really appreciate it and to talk to people about it with passion, this is a game changer. It's a game changer because just the moisture, never think about THC, just moisture, just having the ability to test that A&W repeatedly, not on the full batch of 300 kilos, but like in this small sector of the batch, what was the AW when packaged? Nobody does that. Nobody currently says, when I packaged it, it had this water activity. This is my commitment to you as the consumer. They're not doing it because they say, oh, the cost, the logistics. Hey, we're in the year 2021. It's no longer exactly the case. So,
Yeah.
you know, Guy, let me see if we have any questions that we can tackle before we go here. Yes, Javier, yes, absolutely. It's a faster tool. Guy, why don't you give us context for just how fast it provides results? Tell me, how long after I put my sample in do I get a result?
If you run in the quickest setup, it's around 2.5 minutes. The high end setup is 5 minutes. I can tell you now that in the lab we're working on even faster speeds. But compared to an HPLC where you may be waiting for days, it's a no-brainer.
No, Guy, no, days if you're lucky. Guy, most laboratories have a turnaround time. See, if you're not an LP, this is the thing. If you're a consumer, you're a home grower, it might take 3 weeks to get back to you because they're going to prioritize the LPs that are the repeat buyers.
Before
they might get it back in a few days. They might, if they're lucky, sometimes 5 days a week. Okay, so that's for the LP that's got like the really good account on there. Remember, all the smaller ones of us, we're not going to get treated like the LP. So if you're like a breeder, and I can tell you that from firsthand experience, and having an account in laboratories for 10 years, Okay, like I've worked with laboratories like MB for 10 years, and still I have to wait 2 weeks to get my sample back because I'm not a big fish. I'm a breeder that spends my own money on refining my phenotypes. And so I don't submit as many samples as an LP that's going to submit regular. So they don't treat me with the same love. Guy, I can't tell you what the machine, I'll be frank with you, I haven't been using it as much as I would because I haven't been doing a lot of phenotyping in the past 4 months. But I'm getting back into phenotyping rounds right now, and I can't tell you how much easier your machine is gonna make my life. So big thanks to you and the whole team down at GemmaCert for me. You know, big thanks.
You're welcome, and we really need your feedback and everyone, all our users' feedback, because that's how we keep improving the product over time.
We have a good question here that we're gonna close off with.
Okay.
Well, thank you for that. Because the pro version of your machine, I've got the light version that just allows me to test my flower really quickly and easily. But the pro version also does concentrate. Sky, tell me about concentrate testing. How confident are you in the accuracy that it's useful? I haven't used the pro version. We have a question here for one of the extractors. What do you think?
Okay, well, the pro version really does right now does what is the crude, the extract, which our extractors use it basically they use it twice or 3 times even. One is to validate the incoming biomass that it's up to spec. If they want to make a high THC extraction, they want to see the biomass has plenty of THC in it. On the other side of the production or the extraction, they want to make sure that the extraction is delivering the desired results. And extraction is, it's very accurate there because you're talking about a much easier sample to test than a complex flower bud. It's a homogeneous sample. And we do have, by the way, we do have some extractors who are examining the flowers or the biomass a second time and seeing that there's still active THC and CBD in there to do another extraction run instead of throwing that biomass away. So yeah, so that can save you a lot of money.
And what are you seeing in terms of how closely are you following HPLC results? What's your percentage degree of variance? What would you say?
Okay, that's a good question. Assuming that the HPLC is fully calibrated, operated by a skilled technician, everything's perfect.
That's everything.
We will be within 10% of the actual result. So if the actual result is 10% THC, we'll be between 9% and 11% THC. And to make a decision in real time, that's sufficiently accurate.
No, absolutely. That's a thing that I already know. If you know the degree of variance that it has from actual HPLC, you can already, and this is what I always tell people, that even your HPLC result is not to be taken down to the decimal point. That's a ridiculous HPLC reading. When your HPLC gives you reading, you also have to look at the reading the same way as when you get your GemmaCert. It's that 10 is also potentially 9 or 11.
Could be very,
absolutely right, Guy. I mean, typically, so when you're doing ring testing, you don't want to see above a 14% standard deviation between your labs, because above 14% is beyond what's stipulated in most pharmacopoeias. But and again, folks, that's just fancy name for a book that gives us all of the standards to follow for testing a plant. Okay, I always, this is the thing, when anybody tells me, oh, well, you know, we can't use spectrometer because it's not that accurate, it's always 1 or 2 points off. I'm like, hell, your GCMS is the same thing, your HPLC is the same thing. How is it different? How is this not a tool that we should be looking at just because it can't provide you with a reading that you're like, oh, this is the word of God, it's written on stone, this is exactly what it is. Testing doesn't work that way in general.
Exactly. And you know, the best example, I mean, again, in other industries where HPLCs are used, if there's a dispute between 2 commercial parties and they need to go to mediation or court, nobody they always will bring 3 different lab results from 3 different labs, and because one lab doesn't mean anything. Okay. And that's, so as you said, HPLC is not the word of God either.
Guy, we're out of time, but it was really an amazing conversation. Thank you so much for your time today. I really hope to bring you and your company back at some point, because we're going to have other subject matter where spectroscopy is going to play into it. So be open to having an invitation to come back, Guy. Okay.
I'll gladly accept again, and thank you very much for this opportunity.
Great talk. Thank you, everyone who attended. We will see you in 1 month back here. Remember, noon PST every first